thanks to the AViD team for inviting me to present some of our work. And so as I mentioned, I'm going to tell you a little bit about how we go about thinking about avoiding drug resistance from the get-go by defining what we call the substrate envelope for and our application of this with viral protease inhibitors. So when we think about drug resistance, from globally, it's more than just the viruses that we're studying now, but it really is every quickly evolving disease or system. So whether we're thinking about viruses or fungal infections or bacterial infections, or even in oncology, if you have a quickly evolving system and you have a person who's ill and they take medications, be they small molecules or even going to biologics, they might start feeling better for a while, but after ongoing therapy, they can feel worse. And this is often due to drug resistance and where the medications are no longer effective. And so these processes occur over and over again in quickly evolving diseases, and not just in viruses and cancer cells, but also in agriculture and herbicides and pesticides. Similar things are happening over and over again. And so how do we approach it? As structural biologists, we are at the interface of structure-based drug design and evolution. And we realize that a lot of drug resistance happens when mutations in the macromolecular drug target allow it to retain function while no longer being efficiently inhibited by the drug. And so in order to thwart that, we really not only need to disrupt the drug target's activity, which is necessary, but it's not sufficient for developing a robust drug that avoids resistance. We really want to focus on how to design drugs that last from the get-go. And so my lab has been studying viral proteases for the last 2.5 decades. We did a lot of work on HIV protease, which I won't be telling you about today, but I'll tell you more about some work we've done with hepatitis C protease and most recently with the SARS-CoV-2. So you get a feeling for what I'm describing and why these have turned out to be really good systems for thinking about and avoiding drug resistance from the get-go. So if you think about drug targets and whatever you're targeting, these are often, but although not always, enzymes. And these enzymes interact with a particular substrate and form a functional complex. And if this is on a disease pathway, the disease progresses and you have an unhappy patient. If you're thinking about drug design approaches, whether we're doing small molecule screening or experimental or computational, if we're lucky, we get a hit. And if we're really lucky, this hit is bioavailable and all those good things. But if it's a quickly evolving disease, the enzyme's able to evolve and evolve resistance. And then the inhibitor doesn't bind so well anymore and it fails, but the enzyme's able to still recognize and process its substrate. So we realized that this act of recognizing and processing substrates was in fact an evolutionary constraint. So if we pay attention to the enzyme substrate complex, and this is especially true in a case where the enzyme recognizes multiple diverse substrates, we have an added constraint that we can use in structure-based drug design to avoid drug resistance by fitting the inhibitor in what we call the substrate envelope. And these inhibitors still have to be potent, they still have to be bioavailable, they still have to be all the things that a good drug is. But by fitting within the substrate envelope, a mutation that impacts them will simultaneously impact the recognition and processing of the substrate. So that's the key evolutionary constraint. And these inhibitors should not only maintain potency against the primary target, but other resistant forms of the enzyme target or other polymorphisms that might exist, because all the ones that are viable have to be able to recognize and process substrates. So if you compare our technique to more traditional methods, there's, although resistance still can occur, the probability is starkly decreased. compared to more traditional methods. And so again, this concept that disrupting the drug target's activity is necessary, but not sufficient for developing a robust drug that avoids resistance is sort of in the mainstay of that. So the way I think about the landscape of drug design is a combination of the protein structure and dynamics, the chemical structure of the inhibitor and its potency, and the sequence diversity that's accessible to the drug target. And today I'm going to be not discussing so much in the dynamic space, but more talking about specifically about the substrate envelope. But my lab works in all of these dimensions and the viral proteases turn out to be really good model systems to thinking about avoiding resistance, because as is this sort of schematic of originally HIV, the viral protease is responsible for cutting a series of sites within the viral polyprotein and allowing viral maturation. So as you in the AViD group well know, this is a really powerful drug target. And as Paxlovid is now with SARS-CoV-2 is the main drug that's available to us. It's the message has come home again. But the evolutionary constraint that these viral proteases have to cleave many diverse sequences is what's key in avoiding resistance. So I'm going to tell you a couple stories. The first one is a story that we did over the last probably 15 years on hepatitis C and hepatitis C protease and looking at resistance there. And hepatitis C protease is a serine protease, and it currently is the target for 3 protease inhibitors that are currently used in combination therapy to treat hepatitis C. I mean, hepatitis C is a pretty remarkable story in that only about 10, 15 years ago, the only treatment really was a liver transplant for people infected with hepatitis C. And now for the majority of people, it's a curable disease within a few weeks' time. So, I mean, that in terms of structure-based drug design, that's a pretty phenomenal story. And I believe all of the combination therapies currently used still involve a protease inhibitor, and these are some of them. So hepatitis C protease, like all viral proteases, is responsible for cleaving a number of sites in the viral polyprotein. As I mentioned before, it's a serine protease. And some work we did over 10 years ago was to solve the substrate crystal structures, or actually the product crystal structures, because it turns out serine proteases can have catalytic activity even in if you mutate some of the active site residues. And we defined the substrate envelope by the superimposing the crystal structures of the products crystal structures and calculating a consensus volume with the intersecting van der Waals volumes of the different crystal structures. And that's shown here in the sort of blue cloud, sort of similar as the cartoon that I showed you earlier. When we look at drug resistance in hepatitis C protease, drug resistance clustered just outside the substrate envelope, where the most primary resistant mutations are circled here in red. And you know, it's just like I showed you the cartoon here with the just outside the substrate envelope. And these resistant mutations are often single site mutations, and it's the simple things like an arginine going to a lysine, an alanine going to a valine or threonine, or this aspartic acid going to an alanine. And so to give you a feel for how this works, here's a drug that got into clinical trials but didn't make it to the market called tenofovir. And you can see how it's protruding outside of the substrate envelope, this bicyclic ring, and it's making a nice aromatic stacking on this arginine. So you can imagine if this arginine becomes a lysine, you'll quickly get resistance. Or if this aspartic acid becomes an alanine, this nice ionic network is broken down, and so you can also get resistance. But this particular compound can tolerate changes at this alanine going to a valine or threonine because there's enough flexibility in the small molecule. The So, very much like the cartoon I showed before. What I do want to point out, though, that this compound has a macrocycle here in that fits well within the substrate envelope, and so that's good. While this is sticking out, it's more problematic. Another compound, this is grazoprevir, which is one of the drugs in the Merck combination therapy. This inhibitor still sticks outside of the substrate envelope, but rather than packing on these residues, this bicyclic group packs on the catalytic triad. So those, the catalytic triad, of course, can't mutate it either, so it's structurally invariant, but it has this large macrocycle that is packing specifically over this little alanine that can mutate to a valine or threonine and cause resistance, or this aspartic acid that can become an alanine and lose packing of this. So, it had some good things in going against the catalytic triad, but some not so good things about forming this macrocycle over residues that are known to be variant. And so, what we'd like to do is pay attention to what interactions are good and what are not so good. And the one thing to point out about grazoprevir is grazoprevir was one of the three compounds that I showed you in the beginning, and I'm not an organic chemist, but you can see that whether it's grazoprevir or voxliprevir or clocaprevir, the differences are relatively minor with the addition of a few fluorine atoms or slightly different chemical architectures, but the overall shape of the compound is very similar, as is the susceptibility to drug resistance. So having this macrocycle that's over the two residues I showed you. So our goal was to take the lessons from tenofovir and grazoprevir and combine them into new chemical moieties where we varied different R groups to stay within the substrate envelope, but retain binding affinity that these inhibitors showed. So when we first did this, we wanted to keep the packing against the catalytic triad, so that bicyclic group that was in grazoprevir. And when we solved the first crystal structure of a compound we made, we were very relieved to see that it retained packing against the catalytic triad, and otherwise fit well within the substrate envelope. But unfortunately, it lost some potency, so we made a whole series of different compounds trying to optimize the interactions. And over time, we were able to fit the envelope better and pack against the protein structure. So our best compounds really contour the protein very nicely. As this one you can see does pretty well, but this trimethyl group is sort of floating over a frustrated pocket that water can't fit, and there's not good van der Waals packing. Whereas this compound is able to contour the protein much more closely and has better affinity in the final. So if we look at a series of compounds that we made, the magenta compound had the best fit and stayed within the envelope and retained potency. And you can see this compared to grazoprevir, where we lost about 250-fold resistance. Our other compounds lost much less resistance, and with the most potent compounds losing only about 10 or 12-fold against the resistant mutation of D168A. So this really shows sort of proof of pudding that we were able to design inhibitors that fit within the envelope and retain potency. And our next generation of compounds that we just recently published, we were able to add, again, some fluorine atoms that added other potency. And in viral cell culture, we're able to, we lose less than, we're more potent than some of the drugs that are in the clinic against resistant variants, and similar potency against the wild type. So, the idea of the substrate envelope can both define resistance, but also provide the guide for avoiding resistance while retaining potency. So I'm going to move on to talking about SARS-CoV-2, and this audience needs really no introduction to the M-Pro and TL-Pro, the viral proteases of the coronaviruses. As you know, they're both cysteine proteases, and the two enzymes together cleave 14 different sites in the viral polyprotein. My group has been focusing, as many labs, on SARS-CoV-2 M-Pro, and in terms of looking at substrate specificity, like all viral proteases, the different substrates have very different sequences, with the exception of the glutamine at the P1 site. So the sequence diversity is very, very broad. So we went about in the beginning of the pandemic solving crystal structures of an inactive variant of the M-Pro, where we mutated the catalytic cysteine to an alanine, and we solved crystal structures of 10-mer peptides corresponding to the different cleavage sites, and we were able to solve 9 of the 11 different substrates. This was a collaboration with the Novartis team, and we were able to get nice structures, be able to detail conserved water structure of these different complexes, and really understand the different interactions of the different substrates and what's conserved. And so what we often do is map the van der Waals interactions, and this is the mapping and the extent and the conservation of van der Waals contacts with the different substrates. So the most extensive interactions were with Q189, but there was also substantial interactions with M165 and E166 between the different substrate structures that we saw. If we flip it on its head and look at the interactions with the substrates, not surprisingly, the conserved glutamine at P1, it makes the most extensive interactions of all the substrates, with the P2 and the P4 residues making the next most conserved interactions. We calculated, as we did with the hepatitis C and previously with HIV protease, the substrate envelope from the different substrate crystal structures, and you can see that it's much more conserved on the unprimed side of the cleavage site than on the primed side. Here's another representation of this as sort of a heat map, looking at the conservation of the substrate envelope. And then we looked at how the different inhibitors fit within the substrate envelope. So looking at niraparib, we looked at the crystal structure of that, and fit it within the substrate envelope, and observed where it protruded outside of the substrate envelope. And so in this pinkish color is our contacts of the inhibitor, so you can compare it with the substrate envelope, and you can see what atoms are protruding with the white spheres. To further this, the interactions, so going back, there are interactions with where it's protruding out at glutamine 189, a little bit at methionine 49, and at E166 and asparagine 142. And then when we map the extent of the van der Waals contacts of the inhibitor, you can see that the interactions at 165, 166 are the most extensive, and 189 is also extensive. And here again, it's highlighting it and the substrate envelope without the surface. So you can see the interactions at the 4 residues that I mentioned before, where it's protruding out. The most extensive where it's protruding out is at the E166, but to a lesser extent with these other residues. So what was heartening was then when our, in collaboration with Daniel Boland's lab, who's spoken in this forum earlier, we they did a saturating mutagenesis of the MPRO and found that these residues where the inhibitor was protruding from the substrate envelope all tolerated extensive mutations. And even further, the more recent paper by David Ho et al. Showed that E166B was a primary resistant mutation to nirmatrelvir. I still can't pronounce it after the year, sorry. But so, I mean, we're really able to map out where potential resistance is going to be by paying attention to how the enzyme looks at the substrate, and mapping where inhibitors protrude from the substrate and how extensively they contact the van der Waals surfaces is really important as we look at new compounds, both within your collaboration with the AViDD group, but and other private companies, and seeing that the different protease inhibitors actually map to different residues and to interact with their van der Waals contacts are to a greater or lesser extent. And I think as we think about targeting MPRO, both with SARS-CoV-2 and other viral coronaviruses, we need to think about also the conservation. So there's a lot of variation if we just look at MPROs between different coronaviruses when we go to different subtypes of this viral family. Only a very few residues actually here highlighted in red and orange are either completely conserved or fairly conserved. The rest are variable. And so this much variability is something that we as a community do need to pay attention to. And if we zoom in on the active site, there's a lot of variability even within the active site. So if we're thinking about taking it to the next step and really looking at pan-viral inhibition, we need to pay attention to this. And E166, which is interesting, is completely conserved between the different variant coronaviruses, but it was selected for resistance by Ho et al. and shown by Julia Flynn and Daniel Boland's lab also to be able to tolerate mutation. So these leave open questions of what's conserved evolutionarily might not map out completely what's conserved when under the selective pressure of an inhibitor. So we need to pay attention. And this is further emphasized by some recent work we've done looking at some other species of coronaviruses. We've recently crystallized structures of coronaviruses from beluga whale and from porcine, and we can compare how inhibitors fit with SARS-CoV-2 with each of these and see that the extent of the van der Waals contact does vary from species to species, which makes sense because the residues also are varying within the active site. So with that, I'll just summarize. I hope I've convinced you that the incorporation of the substrate envelope decreases the probability of resistance, and it's possible to design potent inhibitors that fit within the substrate envelope. You're not limited to the potency of substrates by any means. And that we can anticipate resistance, as we did with the once again with the SARS-CoV-2 MPRO analysis. And I think just taking a quick step back to avoid resistance in design, we have to really realize that it's multidimensional and interdependent. And if you think about your drug target interaction, you need to think, if you change your drug, what the modification of the drug implies throughout the target. It may not just be a local effect. Change, a mutation in the active site can impact the drug binding, but also can propagate through the enzyme. But distal changes can also have an effect on binding affinity. And in reality, it's a combination of active site changes in the drug, and distal changes that we have to sort of integrate together and incorporate into a more comprehensive strategy for drug design. Because solving resistance is sort of one step in a path towards thinking about pan-viral inhibition. And with that, I would like to thank my colleagues who've done much of the work that I described today, both current and past, and the funding agencies. The MPRO work was funded by Novartis. The COVID work was funded by so MPRO, the hepatitis C work was funded by NIAID, and others parts of our drug design strategies is funded by NIGMS. Thanks very much, and I'm happy to answer some questions.